Welcome to the Deep Dive, where we crack open complex ideas to get you quickly informed. Today, we're diving into something absolutely fundamental to life on land, how a plant gets its start. We're talking about the incredible journey from a tiny, often dry, dormant seed to a resilient young seedling pushing up towards the light.
That's right. We're exploring the intricate processes outlined in a key source, a chapter from the 6th edition of Plant Physiology and Development. It's a treasure trove detailing everything from seed structure and how they survive tough times to the amazing ways young plants navigate their environment.
Our mission is to guide you through this vital life cycle stage, pulling out the most fascinating insights about how seeds wake up, mobilize their energy, and, orient themselves in a complex world. Think of this as your essential briefing on the strategies plants use and their crucial first steps. Okay, let's unpack this. Before anything else can happen, what exactly is a seed at its core? Our source kicks us off with the basic structure, focusing on flowering plants, which are so important for us.
At its simplest, a seed is basically a protective vessel for a plant embryo. It's got an outer layer, the testa or seed coat, for protection. Now, here's a fun bit of plant trivia right away. Sometimes what we call a seed, like say a grain of wheat or a nut, is actually a fruit. The seed coat is fused to the fruit wall in those cases. Things like beans and peas, though, those are true seeds inside a fruit, the pod.
Uh-huh. A little botanical nuance. Okay, so inside this protective coat is the embryo.
Yeah, the embryo. And it's remarkably simple at this stage. It has an axis that will form the main stem and root, and one or two cotyledons.
The cotyledons.
Yeah.
Those are like the first leaves.
Sort of embryonic leaves, yeah. And often they're packed with stored food. On the axis, you have the tiny embryonic root, the radicle, and the part that will grow into the shoot the stem and leaves called the plumule, and the hypocotyl connects these parts.
And that stored food is non-negotiable, right? They can't just start photosynthesizing immediately.
Absolutely essential. Yeah, these food reserves, starches, proteins, oils, they're stored in different places depending on the seed. Some seeds have a dedicated food storage tissue called the endosperm. Think corn or wheat. Those are endospermic.
Okay.
Others use up the endosperm during development and store the food right in the cotyledons, which get really big, like beans or peas. Those are non-endospermic.
I see.
And then you have some odd cases, like beets, that use a maternal tissue called the perisperm for storage.
Interesting. Now, the source mentions a specific layer in those endospermic seeds, particularly cereals, called the aleurone layer. What's special about that?
Ah, yes, the aleurone layer. This is a really key layer in grains like wheat or barley. It's the outermost part of the endosperm, and it's specialized. Its main job is to churn out digestive enzymes once germination starts.
So it's like the digestive factory.
Pretty much. It's packed with storage proteins too, but its main function is mobilizing those reserves in the endosperm.
Speaking of cereals, they have some unique protective shields, don't they? Adaptations for pushing through soil.
They do, yeah. Very important agriculturally. The embryo has a modified cotyledon called the scutellum. Think of it like a sponge absorbing nutrients from the endosperm.
Okay.
And the young shoot and root have protective sheaths. The coleoptile covers the shoot.
We've got a little helmet.
Exactly. And the coleorhiza protects the emerging root. In maize, there's even something called a mesocotyl that elongates specifically to help push the shoot through the soil surface.
Okay, so the embryo's all packed up, got its food, its protection, but it doesn't necessarily start growing right away. That brings us to the concept of dormancy.
Exactly. Most mature seeds dry out. They enter a sort of resting state called quiescence. If you just add water and conditions are okay, they'll germinate. But dormancy is different. It's an active block to germination, even when conditions seem perfectly fine. It's the seed basically saying, nope, not yet.
Why would a seed do that?
Nope.
Seems counterintuitive.
It's actually a brilliant survival strategy. Dormancy delays germination. This allows time for dispersal, getting carried away by wind or animals. It prevents the seed from sprouting at the wrong time of year, like during a warm spell in autumn, just before winter hits.
Right, that would be bad.
Very bad. And it lets seeds build up in the soil over time, creating what we call a seed bank, just waiting for the right opportunity.
How is this not yet message enforced? Is it like something inside the seed itself or more physical?
It can be both. You can have embryo dormancy, where the block is inherent in the embryo's physiology, its internal state.
Right.
Or it can be coat-imposed dormancy, where the surrounding tissues, the seed coat, or maybe the endosperm are the barrier.
Okay, tell me about coat-imposed dormancy.
Well, it's really varied. The coat can be impermeable to water. That's crucial in dry climates. Think of seeds with really thick, waxy coats. You know that Indian lotus seed that was viable for over 1,000 years? That super tough, waterproof coat was the key. To break this kind of dormancy, you often need scarification, basically, scratching or wearing down the coat, maybe even chemically.
Okay, so water impermeability is one. What else?
The coat or the endosperm can also create mechanical constraint. It can literally be too hard for the tiny root, the radical, to push through, like in tomato seeds.
So it's trapped.
Trapped, exactly. Or the coat can limit gas exchange, restricting the oxygen supply to the embryo. Wild mustard seeds are a good example, much less permeable to oxygen than water.
Huh.
And sometimes the coat traps inhibitory chemicals that the seed itself makes. These chemicals prevent germination until they're leached out, maybe by heavy rain.
So the coat isn't just passive protection. It can be an active gatekeeper.
Precisely. It plays a really active role in timing.
And the other type, embryo dormancy.
Right. That can be physiological, often related to hormone levels, which we'll get to, or it can be morphological. That means the embryo inside the seed just isn't fully developed yet. It actually needs time to grow and mature within the seed before it can even think about emerging. Carrots and celery are examples.
So the seed is released, but the embryo is still under construction, in a way.
Exactly. It needs more development time.
Are there any plants that just skip the whole waiting game? Just germinate right away?
Yes, and it's pretty dramatic. It's called viviparity. That's when the seed germinates while it's still attached to the parent plant.
On the plant?
Wow. Yeah. It's rare in most plants, but essential for some, like the red mangrove growing in tidal zones. The seedling grows quite large on the tree, developing a heavy root structure, so when it drops,
It's ready to stick in the mud.
instantly, it can root almost immediately in that difficult environment.
And we sometimes see something similar in crops, but it's usually a problem there.
Right, that's pre-harvest sprouting. You see it in grains like wheat, especially if there's wet weather during harvest season. It causes huge economic losses.
Not good.
Not good at all. And in maize, you can find mutants that lack key hormone signals, and they'll just germinate prematurely right there on the cob. It really underscores how critical hormonal control is for keeping things in check.
That leads us perfectly to those key chemical messengers. It seems like a classic plant hormone showdown between abscisic acid, ABA, and gibberellin, GA.
It absolutely is. The central idea, the hormone balance theory, proposes that the ratio of ABA to GA is the primary switch determining dormancy versus germination.
Okay, so ABA is the brake, GA is the accelerator.
That's a great way to put it. ABA promotes dormancy, it's the inhibitor. GA promotes germination, breaking dormancy, and allowing growth.
And environmental cues like light or temperature, they tie into this.
They do. They actively shift this ABA-GA balance inside the seed. Or they can change how sensitive the seed is to these hormones. So high ABA levels or low GA levels, or even just being really sensitive to ABA that keeps the seed dormant.
Right.
To break dormancy, the balance has to shift towards higher GA, lower ABA, or maybe increased sensitivity to GA. And this shift is regulated right down at the gene level, controlling the enzymes that make or break down these hormones and affecting proteins that act like, well, like molecular brakes on GA's actions. These are called DeLo proteins.
Okay, so the environment talks to the hormones. How does the real world actually manipulate this hormonal switch to tell the seed it's finally time to grow? What are those environmental signals?
Seeds are amazing integrators. They take in multiple signals, and these cues ultimately converge on tweaking that crucial ABA.GA balance or sensitivity. It's interesting, actually. Often you can just treat a dormant seed with gibberellin, with GA, and it'll germinate. It bypasses the need for the natural environmental cue.
Like a hormonal shortcut.
Exactly. But in nature, key cues include the light, especially for small seeds that might be buried shallowly. This response to light is called photoblasty. There's a light sensor called phytochrome, which we've talked about before in other contexts, that detects red and far red light. The ratio of red to far red tells the seed if it's under shade.
Ah, because leaves filter out red light.
Precisely. So a low red to far red ratio signals shade, and that can inhibit germination. Makes sense, right? Why sprout if you're just going to be immediately shaded out by bigger plants?
Makes perfect sense. What else?
Many seeds, especially from temperate climates, need a period of chilling. We call this stratification, usually cold, wet conditions for weeks or even months.
Like experiencing winter.
Exactly. It prevents them from germinating in the fall, ensuring they wait for spring. It's like a built-in calendar triggered by cold. It also helps synchronize germination within a population.
Clever. Any other triggers?
Yeah, another one is after ripening. This is basically just dry storage at room temperature for a while, weeks or months. It can break dormancy in some species, like barley. Think of seeds produced in spring maturing over the summer heat. That dry period can actually cause ABA levels to decrease.
Huh. Just waiting helps.
Sometimes. And then there are chemical compounds in the environment. Things like nitrate in the soil, often acting together with light, can break dormancy. And chemicals found in smoke, like terraquinolides, are powerful germination triggers for many plants, especially those adapted to fire-prone ecosystems.
Wow. Smoke signals telling seeds to grow.
Literally. It's fascinating how these different signals, light quality, cold, dry storage, specific chemicals, all seem to funnel into manipulating that central ABA.GA switch.
It all comes back to the hormones.
It really does. They've evolved diverse ways to read the environment and essentially tell the seed, okay, the coast is clear, conditions look good, it's time to make a go of it.
So once dormancy is broken and the conditions are right, water's available, temperature's good, the seed transitions into active germination. What does that critical phase look like?
Right, so germination technically starts with the seed taking up water, and it ends when the embryonic root, the radical, actually emerges from the seed coat.
Okay, so radical emergence is the finish line for germination itself.
Correct. Everything that happens after that, as the seedling grows, is called seedling establishment, and water is the absolute first requirement. Dry seeds have incredibly low water content, maybe 5–15%, and metabolism is basically at a standstill. Water uptake reactivates everything.
How does the water get in? Does it just soak in evenly?
It usually happens in 3 phases. Phase I is imbibition, a rapid initial uptake of water. This is mainly physical, driven by water binding to the dry components of the seed. It wakes up basic metabolism like respiration.
Okay, phase 1, rapid soak.
Then comes phase 2. Water uptake slows right down, or even plateaus. But internally, a lot is happening. DNA is being repaired, new proteins are made, the cell's internal structures are getting organized, stored food starts breaking down slightly, which actually helps draw in more water later.
So this is the getting ready phase.
Exactly. The embryo expands a bit, building up pressure, and then the radical pushes through, rupturing the seed coat. That marks the end of germination and the start of phase 3.
And phase 3.
That's when water uptake resumes rapidly again, but this time it's driven by the growth of the seedling itself, cell division and expansion. Full mobilization of stored reserves kicks into high gear now.
So that phase 2, the slow phase, is really where all the crucial internal prep work happens before the root actually breaks out.
Precisely. It's like the seed is powering up all its systems, getting ready for launch, and you can see how vital external water availability is. If the water potential outside the seed is too low, germination just stalls or fails.
Makes sense. Now, once that radical is out, the seedling needs serious energy. It can't photosynthesize yet. Where does that fuel come from?
That's where those massive stores kick in, the carbohydrates, proteins, and lipids packed away in the cotyledons or the endosperm. They become the fuel for growth. Enzymes swing into action. Starches are broken down into sugars by enzymes like amylase and boat amylase. Proteins stored in special vacuoles are broken down into amino acids for building new proteins. Lipids, which are super energy-rich, are processed through complex metabolic pathways.
And that aileron layer in cereals we talked about, this is its moment, right?
This is its star turn. The embryo, specifically the scutellum part, releases gibberellins, GA, into the starchy endosperm. The GA diffuses to the aileron layer and essentially tells those cells, okay, start making and secreting digestive enzymes, especially on amylase, which breaks down starch.
So the embryo is like the command center, sending out the GA signal to the aileron troops to start digesting the rations.
That's a perfect analogy. The enzymes break down the starch into sugars and proteins into amino acids. These soluble nutrients are then absorbed by the scutellum in the embryo and transported to the growing root and shoot tip. Fuel delivery.
How does GA actually do that at the molecular level? How does it turn on those enzyme genes?
It's a need signaling pathway. GA binds to its receptor called GID1. This complex then targets those DELA proteins we mentioned earlier, the brakes. Binding triggers the DELA proteins to be tagged for destruction and broken down by the cell's recycling machinery. Removing the DELA brakes allows other proteins, specifically transcription factors like GAMYB, to become active.
And GAMYB turns on the amylase gene.
Exactly. GAMYB binds to the promoter region of the amylase gene and ramps up its transcription, leading to more enzyme production and secretion.
And ABA, the dormancy hormone, fights against this.
Yes, ABA opposes this GA action. It inhibits the synthesis of ovalase, partly by activating proteins that repress the gene, and partly by preventing the GA-MYB transcription factor from being made. It reinforces that crucial hormonal tug-of-war.
Okay, so the radical is out, the reserves are being mobilized. The plant is now officially a seedling. This seems like a really vulnerable but critical stage seedling establishment. What's key to making it as an independent plant?
Establishment is that huge transition to self-sufficiency. The seedling has to achieve several things. Start effective photosynthesis, get its roots working to absorb water and nutrients, differentiate its tissues properly, and respond effectively to its environment.
Does the size of the original seed matter here?
Absolutely. A larger seed generally means more stored reserves, which gives the seedling a bigger buffer, more time and resources to get itself established before it absolutely has to rely on photosynthesis.
And seedlings emerge differently, right? You mentioned epigeal and hypogeal.
Right. It depends on what happens to the cotyledons. In epigeal germination, like a bean, the hypocotyl elongates and pulls the cotyledons up above the ground. They often turn green and photosynthesize for a bit. In hypogeal germination, like a pea or corn, the cotyledons stay below ground inside the seed coat, and it's the epicotyl, the part above the cotyledons, that elongates to bring the plumule up. The cotyledons just act as a reserve transfer station.
Interesting difference. Now, one of the very first things a seedling must do is figure out which way is up and which way is down and where the light is. This involves those directional growth responses, tropisms.
Absolutely vital. You have gravitropism, the response to gravity. Shoots grow up against gravity, roots grow down with gravity.
Makes sense.
Then phototropism, the response to light. Shoots bend towards a light source.
Got to find that sun.
Exactly. And thigmotropism, the response to touch. This helps roots navigate around obstacles in the soil or allows climbing plants to wrap around supports.
So how do they sense these things, gravity or light, and how does that translate into bending? The source mentions the colony-went hypothesis and lateral auxin redistribution.
That's the core model, yes. It proposes that a directional stimulus gravity pulling downwards, or light hitting one side, causes the plant hormone auxin to be transported unevenly across the growing stem or root tip.
And auxin controls cell elongation.
Primarily, yes. It tells cells how much to stretch. So if you get more auxin on one side...
that side grows differently.
Right. But here's the crucial bit. Shoots and roots respond differently. In a horizontal shoot, gravity causes auxin to accumulate on the lower side. This promotes cell elongation there, so the lower side grows faster, bending the shoot upwards. Same logic for light auxin moves to the shaded side, promotes growth there, bending towards the light.
Okay, more auxin means faster growth in shoots.
But in a horizontal root, auxin also accumulates on the lower side due to gravity. However, roots are much more sensitive to auxin. The concentration that promotes shoot growth actually inhibits root elongation.
Ah, so the lower side of the root grows slower.
Exactly. The upper side elongates more, causing the root to bend downwards following gravity. It's the same hormone, auxin, but the response depends on the tissue and the concentration. Really elegant.
The differential sensitivity is fascinating. And how does auxin actually move directionally like that? You mentioned it's not just diffusion.
No, it's an active, controlled process called polar auxin transport. Auxin is moved from cell to cell in a specific direction, often predominantly downwards in shoots and towards the tip in roots. This requires energy and involves specific protein transporters embedded in the cell membranes. There are influx carriers to bring auxin into a cell, and efflux carriers, particularly the PIN proteins and ABCB transporters that pump auxin out.
And the location of these pumps determines the direction.
Precisely. Where the PIN efflux carriers are located on a cell determines which way the auxin flows next. It creates these directed streams of auxin through the plant tissues.
Okay, back to sensing gravity. How do they know which way is down? You mentioned statoliths earlier.
Right, the starch-statolith hypothesis. This suggests that within specialized cells, called statocytes found in the root cap and in the starch sheath layer of eudicot stems, there are dense organelles filled with starch. These are the statoliths.
Like tiny weights.
Essentially, yes. Being dense, they settle to the bottom of the statocyte cell in response to gravity. It's thought that this physical settling somehow triggers a signal transduction pathway within the cell.
What kind of signal?
It seems to involve rapid changes in things like calcium ion concentration and pH gradients within the cytoplasm. This internal signal then ultimately affects the activity or localization of those p-ion auxin efflux carriers.
So the falling statolith tells the p-ion proteins where to send the auxin.
That's the idea. It redirects auxin flow towards the lower side of the root or stem, initiating the gravitropic bending response.
Okay, and light sensing for phototropism. What's the sensor there?
That's primarily handled by blue light photoreceptors called phototropins, specifically phototropin 1 and 2, located mainly in the very tip of the growing shoot or coleoptile.
And they trigger auxin movement too.
Yes, similar principle. When blue light hits one side of the tip, the phototropins get activated. This activation leads, again through signaling steps, to the lateral redistribution of auxin. Auxin moves away from the illuminated side towards the shaded side,
Promoting growth on the shaded side.
causing the shoot to bend towards the light source. It seems to happen quite quickly, involving modifications to auxin transporters like ABCB19, perhaps inhibiting transport on the light side.
So cool. Now, that transition from growing underground in the dark to suddenly hitting the light is a massive change for a seedling. The source talks about etiolation versus photomorphogenesis.
It's a really dramatic transformation. Seedlings grown in complete darkness are etiolated. They look very different, pale yellow or white because they haven't made chlorophyll, super long and spindly stems, usually with a closed hook at the tip, and tiny undeveloped leaves.
Why do they look like that?
It's all optimized for one thing: reaching light as quickly as possible using the limited stored reserves. The long stem pushes upwards rapidly, the hook protects the delicate shoot tip as it pushes through soil, and energy isn't wasted on expanding leaves yet.
Makes sense. And when they hit light?
They undergo de-etiolation, or photomorphogenesis. It's a whole suite of changes triggered by light perception. The stem elongation slows dramatically, the apical hook opens up, the cotyledons, or first leaves, expand, and importantly, chloroplasts develop and the seedling turns green, getting ready to start photosynthesis.
What triggers this switch?
Photoreceptors again, mainly phytochrome, sensing red and far red light, but also cryptochromes, sensing blue light. Detecting light initiates massive changes in gene expression across the genome.
How do hormones fit into this light response?
Hormones are crucial coordinators. Interestingly, hormones like gibberellins, GA, and brassinosteroids actually promote the etiolated state in the dark. They encourage that rapid stem elongation.
So they suppress photomorphogenesis in the dark.
Exactly. When light is perceived, especially by phytochrome, it leads to reduced levels or reduced sensitivity to GA and brassinosteroids. This removal of the go-go elongation signal allows the seedling to switch to the photomorphogenic pathway, slower stem growth, leaf expansion, greening. You can see this in mutants. Plants deficient in brassinosteroids often look somewhat de-etiolated even when grown in the dark short stems, open cotyledons.
What about that apical hook?
Ethylene is key for maintaining the hook in dark-grown seedlings. Light, particularly red light detected by phytochrome, inhibits ethylene production. Less ethylene causes the hook to open up, exposing the shoot apex. Auxin also seems to play a role in hook formation and maintenance.
And ethylene does other things too, right? Like affecting cell shape.
Yes, at higher concentrations, ethylene often reduces elongation growth but promotes lateral expansion, making stems thicker. This is linked to how ethylene signaling affects the cortical microtubules, the internal scaffolding that guides how cellulose microfibrils are laid down in the cell wall.
Microtubules control the direction of growth.
Essentially, yes. If microtubules are oriented transversely around the cell elongates longitudinally. Ethylene can cause microtubules to reorient more longitudinally, which then directs cellulose deposition that way, restricting elongation and favoring radial swelling or lateral expansion.
Fascinating. Okay, thinking about light again, seedlings don't just respond to the presence of light, they also have to deal with neighbors shading them.
Right, that brings us to shade avoidance. This is a distinct set of responses triggered when a plant detects it's being shaded by other plants, specifically by leaves.
How does it know it's leaf shade?
It's about the quality of the light, not just the quantity. Chlorophyll in leaves absorbs red light very effectively, but lets far red light pass through. So the light filtered through a canopy has a much lower ratio of red light to far red light, RFR ratio, compared to direct sunlight.
And the plant senses this RFR ratio.
Yes, using phytochrome again. Phytochrome exists in two forms. PR absorbs red, and PFR absorbs far red. Direct sunlight, high RFR, keeps most phytochrome in the active PFR form. Shade, low RFR, shifts the balance back towards the inactive PR form.
So low PFR signals shade.
Exactly. And this low PFR signal triggers the shade avoidance response, particularly in plants adapted to open habitats, sun plants. The most obvious response is enhanced stem elongation.
They try to grow taller, faster.
Precisely. They prioritize height growth to try and reach unfiltered sunlight above the canopy, even if it comes at the cost of making fewer branches or smaller leaves. It's a competitive strategy.
How does the low PFR signal cause elongation?
It involves phytochrome interacting with a family of transcription factors called PIFs, phytochrome interacting factors. In the dark or deep shade, low PFR, PIFs are more stable and accumulate. These PIFs then activate genes that promote cell elongation. Hormones like GA are also involved, interacting with the phytochrome PIF system to boost growth.
And this shade avoidance has real-world implications, especially in agriculture.
Absolutely. If you plant crops too densely, they start exhibiting shade avoidance. They put energy into growing tall stems instead of producing grain or fruit, which lowers the overall yield per plant.
So breeders try to reduce this.
Yes, a major success in modern crop breeding, particularly noticeable in maize, has been developing varieties with reduced shade avoidance responses. These shade-tolerant varieties can be planted much more densely without sacrificing individual plant yield as much, leading to significantly higher yields per acre. It's a direct manipulation of this physiological response.
Incredible. Okay, for a seedling to truly establish and become independent, it needs its internal plumbing, the vascular system, and its connection to the soil via roots. How does that develop?
Right. During embryogenesis, the very beginnings of the vascular tissue are laid down as procambium. It's like the undifferentiated precursor tissue. After germination, as the seedling grows, this procambium differentiates into the functional water-conducting xylem and the sugar-transporting phloem. This creates the continuous vascular network connecting roots, stem, and leaves.
What controls this differentiation? Hormones again.
You guessed it. Auxin and cytokinin are crucial players. There are mutants defective in cytokinin signaling or auxin signaling that show severely impaired vascular development. The balance between these two hormones seems critical for patterning the xylem and phloem correctly.
How do scientists study something complex, like making a xylem vessel?
One really useful experimental system involves using zinnia mesophyll cells, basically, leaf cells grown in suspension culture. You can actually induce these cells with the right hormonal cues to differentiate into trachery elements, the main components of xylem.
You can watch a leaf cell turn into a plumbing pipe.
Pretty much. You can observe the whole process. The cells lose their chloroplasts, they lay down thick, patterned secondary cell walls, often reinforced with lignin, and then they undergo programmed cell death, leaving behind the hollow, reinforced tube perfect for water transport. Studies using the system have shown that auxin and cytokinin are needed to kickstart the process, and they've even identified specific molecules, like a proteoglycan called xylogen, that seem to act as signals coordinating the differentiation process between cells.
Amazing. And what about the roots? They need to grow down, anchor the plant, and absorb water and nutrients.
The root tip is a zone of continuous development. Just behind the protective root cap, you have the meristematic zone, where cells divide rapidly. Behind that is the elongation zone, where those new cells expand, pushing the root tip forward through the soil.
And behind that is where they specialize.
Exactly, the differentiation zone or maturation zone. This is where cells mature into their final forms, like vascular tissues, cortex cells, and epidermal cells. And critically, this is where root hairs form.
Ah, the root hairs. Super important for absorption.
Massively important. They are tiny, finger-like extensions of individual epidermal cells that vastly increase the surface area of the root for taking up water and dissolved mineral nutrients.
Do all epidermal cells make root hairs?
No, and the pattern varies between species. In some plants, any epidermal cell can become a root hair. In others, only cells in certain positions relative to the underlying cortical cells become hair-forming cells, trichoblasts, while others remain non-hair cells, atrichoblasts. In Arabidopsis, a model plant, this cell fate decision is made very early in the root tip, controlled by a network of transcription factors responding to positional cues.
And how do the hairs themselves grow?
Root hairs grow by tip growth, similar to how pollen tubes grow. Growth is highly localized at the very tip of the hair, involving targeted delivery of cell wall materials and regulated by things like calcium ion gradients. Hormones, including auxin, ethylene, and others, play significant roles in initiating root hair formation and controlling their final length. Ethylene, for instance, generally promotes root hair growth.
Okay, one last piece of the puzzle, branching. How does a plant build that extensive root system? How are lateral roots made?
Lateral roots, The branches off the main root don't just bud off the surface. They initiate from deep within the primary root, from cells in a specific layer called the pericycle, which surrounds the central vascular cylinder.
So they start from the inside.
Yes. Certain pericycle cells are stimulated to start dividing again. They form a small, organized group of cells, essentially a new root meristem, which then has to push its way outwards through the overlying cortical and epidermal layers of the main root until it emerges.
What triggers this initiation? Is it random?
It doesn't seem to be random. Lateral root initiation is strongly correlated with sites of high auxin concentration within the primary root, often near the xylem poles. It looks like peaks of auxin accumulate periodically along the root, signaling the pericycle cells in those locations.
Okay, time to make a branch here.
So auxin orchestrates root branching too. It plays a really central role, yes. And unlike the primary root, which might have more limited growth in some species, these lateral roots generally have indeterminate growth, meaning they can keep growing and branching themselves, creating that complex soil-exploring network.
Wow, okay, pulling this all together. From a seemingly simple, dry, dormant seed to a photosynthesizing, gravity-sensing, light-tracking seedling with a growing root system. It's just an incredibly complex and beautifully coordinated series of events.
It truly is remarkable. You see how plants constantly integrate internal hormonal signals that ABA-GA balance, auxin gradients, ethylene, cytokinins, brassinosteroids with a whole host of external environmental cues. Water, temperature, light quantity and quality, gravity, touch, even chemicals from smoke or neighboring plants. All these inputs are processed to determine the precise timing, location, and direction of growth needed for survival and establishment. It's a continuous dialogue between the plant and its world.
We've covered so much ground. The different types of dormancy and how seeds break free, the detailed phases of germination and how food reserves are mobilized, especially that aileron layer story. And then how the seedling actually establishes itself, sensing its world through tropisms, responding to light and shade, building its vascular network, and developing its root system with hairs and branches. This deep dive really has pulled out the crucial details from the source text.
Yeah, understanding these fundamental processes isn't just, academically interesting. It has direct practical implications for agriculture, breeding better crops, improving germination, for conservation, understanding how seeds persist and establish in natural environments, and even for understanding how plants might adapt or struggle to adapt to changing environmental conditions.
So what does this all mean?
Well, it really makes you think. Considering this delicate, intricate balance of signals required for a seed just to germinate successfully and for that seedling to establish itself, how vulnerable might these processes be? How might disruptions in just one of these environmental cues, maybe altered temperature patterns affecting stratification, or changing rainfall affecting water availability during germination, or even different light conditions due to invasive species, how might those disruptions impact plant populations and potentially reshape entire ecosystems on a larger scale?
A critical question to ponder for your own explorations. Thanks for joining us for this deep dive.